Okay, first of all, thank the Ion team for having me and allowing me to present the data of two collaborative networks that we started indeed about a year ago. One was focused on BRCA1 design of the AmpliSeq panel, and the other one was focused on colon lung cancer strategies. And also for that purpose, we made use of the custom AmpliSeq panel. Actually, it feels a bit like homecoming for me because back in 1993, I worked here at the University of Florida here, 5 hours up north at Gainesville town. And back in those days, everybody was still sequencing radioactively, and although that was a lot of fun, especially in the wet lab, it was kind of laborious, and you struggled to get 400 bases. And then in the 1990s, or early 1995, we switched to the capillary sequencing, which was a major step forward. But nowadays, we sequence a lot more and a lot faster. And that was although that was a very fruitful setting and a very fruitful way forward, we feel now that also for targeted approaches, the benchtop sequences are probably ready to use. And of course, the Ion Torrent machine, the PGM, is one of the instruments that you can use for that. And that's what we did. About a year ago, we bought 2 machines. And well, we had some harsh demands for this targeted approach, because as soon as you start with a targeted approach, you want to have a robust test, and you want to have, especially if you use FFPE material, the limited amount of DNA available, and you want to be really careful in using that. Also, you want to have very high accuracy, you want to have sensitivity, and of course, you want to have the availability of results within days. And then, of course, there are some technical requirements, flexible. It should be fairly flexible and fairly easy to implement new markers. And of course, you want to detect all mutations in an amplicon that you amplify. This is true for Sanger sequencing, but of course, this is also true for next generation sequencing purposes. With that, we started in the Department of Human Genetics in Nijmegen a collaboration with the Department of Pathology, and that ended up in a new laboratory that was formed, a laboratory of Tumor Genetics. And the first demands that they asked for was a targeted approach for BRCA1 and BRCA2. And then also the Department of Pathology, of course, had a demand for colon and lung cancer panels. And these were the two projects that we started to embark on about roughly half a year ago. And of course, we have some new plans and new ideas, and we're already testing a custom panel for hereditary paragangliomas and pheochromocytomas. We move forward to mismatch repair genes probably within the next few months, and then we'll have some more tests, hopefully at the end of this year, available. So for the BRCA1 and BRCA2 testing, we asked for an AmpliSeq design, and then the Ion Torrent team proposed by the person of Rosella Petraioli to start a collaborative network, and we collaborated with a lab from Portugal, the IPATIMIOP laboratory headed by José Luis Costa, who's in the audience here. And also José Carlos Machado was involved. And with that team, we started to design a test for BRCA1 and BRCA2 testing. I don't know if I have to go through all the details, but as everybody knows, these genes are involved in breast and ovarian cancer. Mutations are dominantly inherited. And the challenging factor is that the combined open reading frame of these genes is about 16 kb. It's highly polymorphic. There are lots of SNPs known within these genes, and it contains quite a few number of homopolymer stretches. The mutations are scattered throughout the gene, so there are not really hotspots available to test for. And most of these mutations that have a clinical relevance are nonsense mutations, frame shift mutation, and indels, but of course also missense mutations are known. As you look at the graphics of the test, you can see that there are two large exons. Exon 11 in both genes is extremely large compared to the others. And that was quite a challenge for the design team to come up with an AmpliSeq panel that was working and had enough overlapping fragments in the design. So why did we switch to the Ion Torrent AmpliSeq approach? At present, we have quite an efficient workflow in the lab, which is fully automated for all the genes that we do in the laboratory. The breast cancer approach contains 84 samples, amplicons, and then, of course, we also perform LPA testing for deletions and insertions. We receive about 50 to 60 samples a month. And with the use of the GSI software, we achieve quite routinely now a turnaround time of 3 weeks. And although that is very efficient, it's still considered quite an expensive technique. So the aim was, with the PGM technology, to at least reduce the cost substantially, to get to even shorter turnaround times, to get a standardized Ion Torrent workflow, and at max 3 multiplex PCR. That was the aim of when we started the project. And we had some harsh demands for the design team of the Ion Torrent AmpliSeq approach. So we wanted to have at least 100% coverage of all coding exons and exon intron boundaries. And we wanted to sequence at least 20 bases, at least 10, but preferably 20 bases into the introns of the two genes. The amplicons should cover all exons, and they should be overlapping if needed in the larger exons. The primers were not allowed to overlap, and there were no validated SNPs allowed in at least the 5 last nucleotides of the primer. And at max, 3 validated SNPs per primer were allowed because of, especially in exon 11, the number of SNPs is quite tremendous, and it's very difficult to find a primer without any SNPs. So then the in silico design was basically designed in collaboration with the team of the Ion Torrent members. And after approval in Nijmegen, the 2 laboratories in Nijmegen and Portugal switched samples and sequenced at least 10 samples to test coverage and to test the workflow. And that turned out to be quite successful. And then we set out to sequence an additional 15 samples to see if the amplicon approach was actually quite efficient in mutation calling. So in total, we sequenced now 65 mutations, and the team is now expanding, so the network is expanding, and we have now an additional 5 other labs who are testing this approach. We have now, in stage 3, sequenced an additional 15 samples in both labs, so in total 65 mutations have been sequenced, and we have never missed a mutation so far. So in 3 multiplex PCR sets that have been designed, they contain about 50 to 55 primer pairs. The different pathogenic mutations that we tested for were also covering homopolymer stretches. They were covering deletions and insertions. They covered point mutations, and that all worked fine. And we're quite convinced that we can also detect exon, full exon deletions, although we're still validating and implementing that in our pipeline. So that's why it's between brackets, but we're fairly confident that we get to that point. And it just showed you some of the one example of what it looks like in SecNext analysis software, one of the software packages that we've tested. And this is a 5 base pair deletion and you can actually see how nice this turns out in this software package. Just to give you a feel on how we stretched the PGM particularly for homopolymer stretches and for indels mutations, because that's where the Achilles heel of the technique is located. So we tested quite a few samples with known mutations in homopolymer stretches. And well, as you can see, the top one is already quite a large homopolymer stretch. And all these mutations were detected by the technology. So now I'd like to switch gears to the Onco network consortium that we started. Also coordinated by Rosella Petraroli from the Ion team. And Enrico was involved for technical assistance. And then 8 other labs, covering most part of Europe, were asked to join. And with this consortium, we started testing the various genes that we wanted to test in this approach. It basically followed the same strategy as the BRCA endeavor. And we optimized the workflow. We validated the custom design for the colon lung cancer panel. It contains 19 amplicons, 22 genes, all involved in colon and lung cancer. And I'd like to stress that we only use 10 nanograms of FFPE material in a single reaction. But I also like to stress that this is not covering the complete genes. It only detects hotspot mutations in this package. So we divided the approach in 3 different stages. 5 control FFPE samples were initially sequenced in the first stage, just to get a feel for the design and to get the workflow going. In stage 2, we set up a blind strategy where we asked the various participating laboratories to send in 10 different tested samples that they had tested previously, and they received back 10 blinded samples for sequencing, not knowing what type of mutation they would detect. And then in stage 3, we sequenced another 19 additional FFPE samples. But the question there was made a bit more difficult because the samples were varying greatly in tumor content and tumor level. So this is a graphical overview of the strategy, showing you that in total we would sequence 155 samples in 180 experiments in 6 different labs. This is the results from stage 1. All the participating laboratories were capable of detecting all the mutations without any major hurdles there. So we felt confident to start the blind study. And then in stage 2, we sent various samples for the various genes for various types of single nucleotide variants, but also insertion deletions were there. And we also introduced just wild-type samples. And in total, all the labs were capable of detecting or not detecting the mutation. So the wild-type variants were properly called, and also the other mutations were properly called. So also this was a success, and then we moved forward to stage 3, where we introduced quite a large variation of tumor content in the samples. And I'd like to point to number 7. Of course, this is the interesting one, because this one was apparently missed. But that was not because of the technology, that was just because there was not enough DNA in there. We tried to confirm that by Sanger sequencing, and also by Sanger sequencing it was just impossible to get a proper sequence. And the other thing I'd like to show you is sample 11. Our software was set at a target level of about 5% mutation frequency. And this sample contained only 2% of mutation frequency. which was missed by the software because of this setting. As soon as we lowered the setting to 2%, the software just called it properly, and just by looking at the data we knew that the mutation was there. So also in this stage all samples were successfully sequenced. And this is just to show you that also amplicon coverage was quite a concern to us, because well, we need about 500x coverage, and of course you notice already that quite a few amplicons have a coverage that was too low, and you lose sensitivity there, and you're not calling these low mutation frequencies reliably. And of course you have also amplicons that have a coverage of 10,000 times, so which is kind of a shame, because you lose capacity of the chip there. So we said to the software, the AmpliSeq developing team, we have to have a new design which is more evenly spread. And instead of 1,000 times coverage, the version 2 of this AmpliSeq panel has now a 10x difference in coverage, showing a much more even spread. And this allowed us to, instead of sequencing 5 samples on a 316 gene chip, this allowed us to sequence 8 samples on a 316 chip, which makes the test, well, you have more capacity in your lab, but also it makes the test cheaper, so that's also a major advantage. The new design was basically retested in a new validation experiment. We again sequenced 8 FFPE samples just to see if the test was capable of detecting all the variants, and then again the same 15 samples were sequenced by all the laboratories, all the other laboratories, to see if also the difficult samples were properly called. And in total we have now sequenced 98 samples in 138 experiments in 6 different labs. So again showing that the technique is quite robust. So coming back to the slide, one of the first slides that I showed you, that the requirements of this test, of a routine molecular test, is that you need a robust performance. Well, I think I've showed you some results there, that even in various different labs, everybody is just capable of getting the proper results. So also the test is highly accurate. It has sensitivity, 5% is easily detected, and you can even go lower than 5%, although that's probably stretching the limits there. Loss of function mutations are properly called, also in the BRCA panel. Results are available within days, and also the technical requirements, like easy to implement and easy to implement new markers, was matched. And also, so far, we have detected all the variants that we tested for. So we are on our way to the implementation of BRCA testing using the custom panel design. We need to implement the workflow into our LIMS system. We surely going to optimize software setting. We haven't set on a real software package, so we're currently testing the various commercial packages available. We would like to validate the combination of the various approaches to get to a more generic workflow. Instead of running a sample and then running another sample, you can probably combine the two, but we haven't touched upon that yet. Of course, we have to finish our standard operational procedures in the lab. Validation reports have been finished. Of course, we're going to start participating in quality schemes, and of course, the other major thing we have to now do is train technicians and personnel. And this is what the laboratory workflow would look like, especially for the Department of Pathology. So we would basically run 3 days a week the various tests in the lab. So probably that will be on Monday, on Wednesday, and on Friday. And then they can use the other days to do the DNA isolation, and the other day you can do the interpretation and the final reporting. So turnaround times we would like to see go down to at least 3 to 5 days. So with that, I'd like to acknowledge all the participating members of the two consortia, and I'll take questions afterwards.
Thank you, Marcel.